In this last video for Unit 2, we're going to be talking about some of the routine ABO testing that we would perform in blood bank. So the ABO grouping is the most frequently performed test in blood bank. So that's just us determining the patient's blood type. These basic procedures in immunohematology are derived from the principle of placing a source of antigen and a source of antibody into a testing environment to detect an antigen-antibody reaction. Both ABO forward and reverse grouping tests have to be performed on all donors and patients. There's always an inverse reciprocal relationship between the forward and reverse types, so it helps serve as a check. One serves as a check on the other. So for example, if an A person has, or if an individual has A antigens on their red cells, there should be anti-B antibodies in their serum since this patient lacks the B antigen. So that should be represented in their forward and reverse type. Patients that are scheduled for surgery or obstetric patients should always have a recent type and screen on file. The reason for this is that it helps for quicker antibody identification. If the patient were to need a blood transfusion, it helps us perform a quicker cross match, meaning that we've found compatible blood for transfusion. And you can have matching donor units on hand if that patient were to need a transfusion. Okay, so the forward grouping, which is sometimes referred to as the front type, is defined as using a known source of commercial antisera. So that means that the reagent that you're using in the forward type is either anti-B, anti-B, anti-D, and we use those antisera to detect antigens on the patient's red cells. So the principle of the forward testing, we're using known antibodies to detect unknown antigens. The known antibodies are in our reagents. The unknown antigens are on our patient's red cells. So for example, to detect the presence of the B antigen on a patient's red cells, patient's red cells are combined with a commercial source of antibody, so that's our anti-B reagent. And if the B antigen is present on the patient's cells, then agglutination is going to be observed in the test tube. So forward typing includes the patient's blood type and their RH status. Weak D typing, for this you have to incubate both negative tubes from the D type. So your patient test and the control at 37 degrees Celsius for 15 minutes. You have to wash both tubes 3 times. Then we add 2 drops of monoclonal AHG reagent. We spin and we read the results. and really in the normal blood bank setting, we're not going to perform, the only patients that we're going to perform a weak D on are babies, or if the doctor were to request a weak D. And then reverse grouping, also referred to as the back type, is defined as detecting ABO antibodies in the patient's serum by using known reagent red cells. So that would be our A1 and B cells. So the principle of the back type, we're using known antigens. So antigens are on the red cells. So we're using commercially prepared reagent red cells to detect unknown antibodies. So we're trying to determine if the patient has antibodies. So an example of this would be to detect the presence of anti-B in a patient's serum. The patient's serum is combined with a commercial source of antigen, which is our B cells. So the back type includes only the patient's ABO back type. It does not include an RH. And if you remember from one of the last videos, we said the ABO antibodies are naturally occurring. So every person should have naturally occurring ABO antibodies, but the only way that they would have Rh antibodies is if they had been exposed either through pregnancy or transfusion. Okay, the antibody screen. So we use this, the screen, to detect antibodies on the patient's red cell antigens. So it has to be performed on a patient before they receive a transfusion. So if the antibody screen is positive, the units to be transfused also have to be screened, and they have to be negative for the antigens that the patient has the antibodies for. The screen would also be performed prior to surgical procedures in order to have matching blood on hand if needed. This also allows for a quicker cross match and quicker antibody ID. So if the patient, what we're looking for in the antibody screen are antibodies outside of the normal naturally occurring ABO antibodies. So we're trying to determine if this patient has, at any point in the past, developed these allo antibodies in response to either pregnancy or transfusion, because if they do have these unexpected antibodies, and they require a blood transfusion, we have to make sure to give them blood that is compatible, that they're not going to have reactivity to. Okay, the cross match determines the serologic compatibility between the donor and the patient before transfusion. So in the cross match, so we've decided, or the doctor has decided, the patient needs transfused blood. We have to cross match the donor unit of red cells with the patient's serum or plasma to make sure that they are compatible. An antiglobulin test detects IgG molecules and complement protein molecules that have attached to red cells but have not yet resulted in a visible agglutination reaction. So this can be done with, hold on just one second, can be done with an indirect antiglobulin test, also referred to as an IAT. So it detects the antibody bound to red cells in vitro. A positive IAT indicates a specific reaction between an antibody in the plasma and an antigen present on the red cells. So this means that the red cells have been sensitized. So it detects incomplete or non-agglutinating antibodies to potential donor red cells. So again, this is called compatibility testing, or the cross match, or it can also be done to screen cells, your antibody screen, in plasma. So the IAT is a two-step procedure. It requires an incubation step. That incubation step serves to encourage the sensitization of the cells, and then it also includes a wash stage. So this allows time for antibody attachment to the red cell antigen. It removes any unbound antibody or complement proteins that are left in the plasma. After washing, the second step is when we add our AHG reagent, which is used to enhance that lattice formation. So if you remember from Unit 1, the two steps in this process are the sensitization of the red cells and then the lattice formation, where they actually, the red cells actually join together. So you add the AHG reagent to the test, then centrifuge your sample. A positive result is agglutination. So several blood bank tests incorporate an indirect antiglobulin reaction phase in their procedures. So that includes your antibody screens, antibody identification, cross-matching, and antigen typing. Okay, some different sources of error. False negatives can result if the cells are not washed adequately. So that wash stage is critical to remove any unbound antibody or complement. So if this is not done, those unbound antibodies can neutralize the reagent, and it would mask a true positive result. false negatives can also result from if there's a delay between the wash stage and adding the AHG reagent. If weak reactions are overlooked, if the tech forgets to add the AHG, if the sample is under centrifuged, or if the tech has prepared an inappropriate cell suspension. So we also mentioned previously that cell suspension should be 2 to 5% because we want these reactions to occur within the zone of equivalence. So if that cell suspension is not correct, it could lead to false negatives. Okay, false positives can come from if the glassware that you're using in your testing procedures is dirty. Some particulate matter on the glassware could be seen as agglutination. If the sample is over-centrifuged, if there are microclots in the specimen, or if the cells were already agglutinated before the wash stage. Okay, and I think, so the antibody screening, the purpose again is to detect antibodies with specificity to the patient's to red cell antigens. Antibody identification, or antibody ID, identifies the specificity of our red cell antibodies. So the screening process determines if the patient has antibodies. The antibody ID determines the specificity of those antibodies. The cross-match is used to determine compatibility between donor red cells and the patient before transfusion. And then antigen typing identifies a specific red cell antigen either in a patient or a donor. Okay. The direct antiglobulin test, or DAT, detects antibodies bound to red cells in vivo. So IAT is something that we're doing in, we're kind of inducing that reaction in a test tube. The DAT detects antibodies that have been bound in the patient's body. So it does not happen under normal circumstances, but it can be a consequence of some clinical events. So like if the patient has warm or cold autoimmune hemolytic anemia, hemolytic disease of the fetus and newborn, there are drug-related mechanisms that can cause this, or an antibody reaction to transfused red cells. So a positive DAT is an important indicator of potential immune-mediated red cell destruction in the body. So this goes back to some of the concepts that we talked about in Unit 1. Your body is recognizing something as foreign, and it's going to kind of attack. In the DAT, there is no incubation or wash stage necessary because that those antigen antibody complexes have already been formed in vivo. Okay, so the DAT procedure. So the patient sample that we're going to use is EDTA blood. We want a fresh sample from the patient. We don't want it to be refrigerated, and it can't be too old of a sample. So the patient's red cells are washed at least 3 times to remove any unbound proteins. We add our AHG reagent. Agglutination with polyspecific reagent means that the patient's cells are coated either with IgG antibody or complement, or both. Agglutination with monoclonal reagent, which could be just anti-IgG, indicates that the polyclonal tube was positive because of IgG antibody. Agglutination with anti-C3b or C3d reagents indicates that complement is on the patient's cells. So you would start with polyspecific or polyclonal reagent, which tests for both IgG and complement. So if it's positive, then you would go on and test monoclonal reagents to test for IgG and complement. An autocontrol is patient's washed cells and saline. So it gets tested also to be sure that the patient's cells are not spontaneously agglutinating without those AHG reagents. And the patient's history and clinical condition should be taken into account if the DAT is performed. There is a video in Moodle on the DAT procedure. Okay, so in the case, these are examples of situations that could cause a positive DAT. So transfusion reaction. In that case, the donor cells are coated with IgG. And the source of that IgG is that the patient, the recipient who has received the transfusion, has antibody present in their serum that is reacting with the donor cells. In the case of hemolytic disease of the fetus and newborn, you've got fetal cells that are coated with IgG. And the source of that IgG is maternal antibody that has crossed the placenta. So with HDFN, you've got mom who is Rh negative, baby who is Rh positive. Mom is producing antibodies to that D antigen that the baby has, and then the antibodies that mom produces can attack baby's cells. Autoimmune hemolytic anemia can be due to IgG or C3 or both on the patient's red cells, the source of which would be a patient autoantibody. And then in drug-related mechanisms, this could be an IgG drug complex that's attached to the cells. So it's just an immune complex that's formed with that drug. And then some differences in DAT versus IAT. So your DAT detects IgG in complement-coated red cells. IAT detects IgG in complement-coated red cells. The DAT, IgG attachment to the red cells, has already occurred within the patient's body, so we're just trying to determine if that is what has happened. In the IAT, that IgG attachment to the red cells occurs during the incubation step, so it occurs during our testing. DAT is one step, IAT is two step. So remember, it's got that incubation, wash step, and then the addition of the AHG. In the DAT, the patient's red cells are tested with antiglobulin reagent, so that's your AHG, without that incubation step. But in the IAT, it requires that incubation step before the addition of the AHG, and you can use antibody enhancement media also. So your DAT is testing to determine if some of these clinical conditions have occurred, either HDFN, hemolytic transfusion reaction, autoimmune hemolytic anemia. The IAT is used as a reaction phase for different tests, such as the antibody screen or an antibody identification panel. And I think I skipped over. Well, maybe not. Oh yeah, adsorption and elution. So an adsorption is a procedure that uses known red cell antigens to remove red cell antibodies from a solution. So we're trying to remove antibodies from either plasma or antisera. So for example, if you've got a group A red cells that can remove anti-A from a solution, and the reason why we would want to do that is if we suspect multiple antibodies, we can get rid of one to help us easier be able to identify the other one. And then an elution is a procedure that dissociates the antigen-antibody complexes on the red cell. So you've got, once you've dissociated that antigen and antibody, you can test that freed IgG antibody for specificity. So an elution can be performed in a few different ways. There's a heat or thawing method, and then there's a couple others beyond that that we won't really go into. Okay, so then getting back to here. So those routine procedures performed in blood bank, ABO, which again is your forward grouping, and the D-typing. is used to detect A, B, and D antigens. So we're trying to determine the patient's blood type. We're testing the patient's red cells, because that's where the antigens are, against a commercial source of anti-A, anti-B, and anti-D. So that's the source of the antibody. That's our reagents. So the reverse type, we're looking for, does the patient have antibodies in their serum? So the source of the antigen is the reverse grouping cells, so A1 and B cells, so those are our reagents. And then we're testing it against the patient's serum or plasma to look for antibodies that they have in their serum or plasma. Antibody screen detects antibodies with specificity to red cell antigens. So our screening cells, which come as a packaged set of cells, so red cells, are what we're testing against the patient's serum or plasma to look for or to determine if the patient has any allo antibodies. So at this point, we're not testing for the naturally occurring ABO. We're testing to see if this patient has any antibodies as the result of either pregnancy or transfusion. The antibody screen, if it's positive, will lead us to antibody identification. So this is sometimes referred to as like a full panel or antibody panel. Helps us to identify the specificity of the antibodies that were detected in the screen. So we're using panel cells, and it's really just the same thing as our screening cells, but just an extended number of them. We're still testing against the patient's serum or plasma because that is where the antibody is that we're trying to identify. And then the cross match helps us to determine compatibility between donor red cells and the patient before transfusion. So we're testing the donor red cells because that's the source of the antigen. We're testing it against the patient's serum or plasma because if they have antibodies, that's where they're going to be located. Okay, talking about some different blood banking reagents. So quality control. So just like we talked about in micro, we have to do QC in every department of the lab for every test that we perform. So checks on blood banking reagents and equipment we have to perform before we use the equipment or before we use the reagents to test on patient or donor samples. Those daily tasks, quality control tasks that we have to perform are based on regulations, accreditation, manufacturers' product inserts, state and local requirements to help us determine if the reagents meet preset acceptable performance criteria. The potency of the reactions have to meet standards and they have to be recorded, and those records are going to be reviewed periodically. So for example, if you've got an anti-A reagent tested against reagent red cells that are known to possess the A antigen, you should get an agglutination reaction of either 3+ or 4+ for optimal reagent performance. So if you're getting an agglutination reaction that's less than 3+ in strength with your group A red cells, the potency of the anti-A reagent may be deteriorating. So if you're not getting the reactions that you expect, then you might need to get some new reagents. Okay, polyclonal versus monoclonal antibody reagents. So I kind of touched on this a minute ago. Polyclonal reagents are made from several different clones of B cells that secrete antibody of different species. So this is when we were talking about DAT, we mentioned polyclonal DAT, which is made to detect either IgG or complement. Monoclonal is made from single clones of B cells that secrete antibodies of the same specificity. So these monoclonal reagents have largely replaced the polyclonal antisera as routine blood grouping reagents. But we do still use monoclonal for the IAT or the DAT to determine if the difference between the IgG or the complement. And then here's just here's that same information in a table. Okay, your reagent red cells. So these are known red cell antigens that we use for the purpose of detecting antibodies. So A1 cells and B cells are our reagent red cells used in the BAC type to detect antibodies that might be present in the patient's serum. Antisera are known red cell antibodies. So these are commercially prepared anti-A, anti-B, and anti-D reagents that we use to detect the corresponding antigens in the patient's red cell suspension, which again has to be a 2 to 5% suspension. And we create that 2 to 5% suspension by mixing the patient's red cells with blood bank saline. So this is what we use in the forward type. Anti-D is used for the determination of the patient's Rh status, which is done in the forward type only. It used to be high protein and low protein varieties of your anti-D reagent, but now it's mostly low protein version with a similar protein concentration to your ABO typing reagents, which uses a 6% bovine albumin solution. If a high protein reagent is used, an Rh control is required to control for false positives caused by additives that could be in the diluent. Your anti-D reagent can be IgM or IgG or a blend, which helps to detect weak D antigen. If a monoclonal reagent is used, a saline control, which is just your patient cells, and saline is used if the patient types as AB positive, just to make sure that it is not a false reaction. Okay, our screening cells. So these are the reagents used in the antibody screen that looks for, they're used to detect antibodies with specificity to red cell antigens in the patient and donor samples. The reagent red cells come from group O donors, and this is because group O individuals lack the A and B antigens. And these screening sets will come either as a 2 vial or a 3 vial set. They're available as a set that is derived from a single donor or from 2 donors in equal proportions. And these vials are made to test for antibodies performed on recipient specimens. Pooled reagents can be used to test donor samples. So each lot of screen cells comes with the antigenic profile, which is, this is what it looks like. It's referred to as an antigram of each donor. So screening cells that are licensed by the FDA require antigenic profiles capable of detecting the most clinically significant red cell antibodies. So these are considered the most clinically significant red cell antibodies. We've talked about the Rh blood group system. We're going to talk about MNS and all these others in the next unit. But you can see, so this was one donor provided this screen cell sample, and this was this donor's phenotype. So you can see all of the cells that the patient was positive and negative for. This is a second donor that helps make up they're making up the second reagent vial in this screening set. And you can see their profile. So what they're doing is they're looking for one that's maybe positive, and so we can get positive and negative reactions for each of these antibodies. And then the third, if it was a 3 vial screening set, you would have one that was D negative. Okay, panel cells, also referred to as antibody panel cells, determine the specificity of a red cell antibody that the antibody screen has detected. So we start with the antibody screen, which is our 2 or 3 vial set, to determine if the patient has an antibody. We're doing the panel cells to identify that what that antibody is. So again, those panel cells are made up of donor cells from group O individuals. They get packaged in sets of 10 or more. So whereas those screening cells are 2 or 3 vials, the panel cells are going to be 10 or more vials. You've got an antigram included with that also. Okay, AHG, which stands for anti-human globulin, is used in the IAT and the DAT procedures. You can use mono or polyspecific reagents to detect either IgG or IgG in complement. If agglutination occurs in either the IAT or the DAT procedure, that means that either IgG or complement is present. So then you would have to retest with your monoclonal reagents to determine if that reaction is positive due to IgG or C3d or C3b. AHG reagent that we use is produced by injecting animals, and it's usually either mice or rabbits, with human antigens so that the animals produce anti-human antibodies. Okay, IgG sensitized red cells, which are more commonly known as CHEK cells, are used as a control system for the antiglobulin tests that get interpreted as negative. So we add our CHEK cells to negative AHG reactions, and we should get a positive agglutination reaction after the tube is centrifuged. So if there is no agglutination, we want to repeat testing. So our CHEK cells are just a check. It can detect false negative reactions due to failure to add AHG, failure of the AHG reagent to react accordingly, or failure to adequately wash the cells. So it's just kind of a double check on our procedure. So we add CHEK cells to all negative AHG reactions, and the results should be positive. Okay, antibody potentiators and lectins. So these are commercially available reagents that enhance the detection of IgG by increasing their reactivity. So we have enhancement media. So these are reagents that enhance or speed up the antibody antigen reaction. And it does this by a couple different methods, either by reducing the zeta potential of the red cell membrane by adjusting the in vitro testing environment to promote agglutination. And then we have antibody potentiators. So these are reagents or methods that enhance or speed up the antigen-antibody reaction. So it could enhance antibody uptake, promote direct agglutination, or both. So this also is done most commonly by reducing the zeta potential. So some of the major types of potentiators that we use are low ionic strength saline, which is more commonly referred to as LIS, which increases the rate of antibody uptake, reduces the zeta potential. It also reduces the incubation time to 10 minutes. So if you're using LIS, you will have a decreased incubation time in your process, and this speeds up the agglutination. Polyethylene glycol, also referred to as PEG, concentrates the antibody in the test mixture while creating a low ionic environment that enhances the rate of antibody uptake. This one can enhance warm and cold autoantibodies. It removes water molecules. Incubation time if you're using this potentiator is 15 minutes. Bovine serum albumin, or BSA, reduces the repulsion between the cells, but it doesn't shorten the incubation time. And I think the normal incubation time, if you're not using a potentiator, is 30 minutes. And then proteolytic enzymes such as physin, papain, and bromelain remove the negative charges from the red cell membrane, which reduces the zeta potential. It denatures some red cell antigens, so it's not usually used as a potentiator in the antibody screen because it can eliminate some of the antigens from the red cells. So this one can be used as an additional tool for investigating complex antibody problems. So if you get to a point where you realize that maybe the patient has multiple antibodies and you need to enhance some or rule out others, this is just kind of an extra tool that you can use to do that. Okay, your lectins are plant extracts that are useful to use in blood bank reagents. They bind to the carbohydrate portion of certain red cell antigens, and they can agglutinate the red cells. So they can be helpful in identifying antigens present on patient or donor red cells. They were first discovered in plants, but they've also been found in bacteria and mammals. Dolichos biflorus is the lectin that we use to create the A1 antigen. Ulex europaeus is specific for the H antigen. Vicia graminia is specific for the N antigen, and Iberis amara is specific for the M antigen. Okay, and autocontrol. Autocontrol is not a reagent again, but it's a test that would be done in conjunction with certain patient tests to serve as a control. So you would do an autocontrol with an antibody screen. And what you're doing is testing patient serum or plasma and the patient's red cells and whatever potentiator you used in the antibody screen. If the autocontrol is not tested at the time of the screen, then a DAT should be performed if the screen is positive. And that just helps you to determine if reactivity is coming from the patient's cells or from reactivity with your reagents. Okay, some alternative testing methods to the tube test. Microplate testing. So you've got a plate with multiple testing wells. So each one of those wells is just similar. It's like a short test tube. So the same principles that apply to agglutination in the test tubes also apply to the testing in these microplates. So it can be adapted to red cell antigen testing or serum testing for antibody detection. So you've got small quantities of red cells and antisera are added to the wells. The plates get centrifuged. Cell buttons are resuspended by a mechanical shaker inside the instrument. Then you've got a concentrated button that is indicative of an antigen-antibody reaction. And then the instrument is going to have a photometric device that reads the reactions and displays them on the computer screen. So your reactions, it's the same concept. What's happening in these little teeny tiny wells are the same as what would happen if you were doing the tube testing on the bench. So the reactions are going to be the same in the microplate testing as it would be in the tube testing. So that's what those microplate wells look like. Okay, gel technology uses dextran acrylamide gel particles to trap agglutinated red cells, provides objective and consistent interpretations of agglutination. But this one is this one's backwards. So if you're using a gel method, you kind of have to get used to it. So a positive reaction is will show as agglutinated red cells trapped in the gel at various levels. So that indicates your different strength of reactions from 1+ to 4+. Negative means that there's a button on the bottom of the microtube. So right there. So it's backwards from the tube from the tube method because you've got the button at the bottom that indicates a negative reaction, and then your positive reactions, you've got the button that's going to be trapped at different levels in the gel. Okay, solid phase testing uses microplate test wells with immobilized reagent red cells. So the major advantages of this testing method includes standardization, stable, defined endpoints that lead to more objective and consistent interpretations of those agglutination reactions. And again, the reactions are opposite of those that you would see in tube testing. This testing can be used for antibody screens, antibody identification, and compatibility testing. In a direct test, the antibody is fixed to the wells. So antigen positive red cells from the patient are going to adhere to the sides and bottom of the wells because that's where the antibody is fixed to. Antigen negative red cells from the patient are going to settle to the bottom of the well, form a red cell button after centrifugation. In an indirect test, you've got red cell membranes that are bound to the wells, and unknown patient serum is added and allowed to react. So this allows the capture of IgG antibodies from the patient serum by the red cell membranes. And then you've got a wash step that removes unbound IgG antibodies, followed by the addition of your IgG coated indicator red cells. In a positive indirect test, the indicator cells adhere to the sides and bottom of the well. In a negative indirect test, the indicator cells settle to the bottom of the wells and form a red cell button after centrifugation. So again, that response is opposite of what you would expect to see in tube testing. There is a video in Moodle on gel testing. Okay, some different ABO discrepancies that can occur. So this is when if it's an ABO discrepancy, and a discrepancy just means that you're getting test results that you do not expect. So with an ABO discrepancy, you've got an ABO phenotype where the results of the red cell testing do not agree with the results of the expected serum testing. So that just means your forward and your reverse type don't correspond. So it may be indicated when some of the following observations are noted in the results of the ABO testing. So if you've got agglutination strengths of the typing reactions are weaker than expected. So usually reactions in ABO red cell testing with your anti-A and your anti-B reagents are going to be strong reactions, so 3+ or 4+. The results of ABO serum testing with your reagent A1 and B cells are 2+ to 4+. So if you've got weaker reactions than expected. So if your expected reactions in your ABO red cell testing and serum testing are missing. So for example, you've got a group O individual that's missing one or both of the reactions in the serum testing, which is your reverse type, with your reagent A1 and B cells. Or if you've got extra reactions that are noted in either the ABO red cell or the serum test. So you've either got weaker reactions in the forward or reverse, you've got missing reactions in the forward or the reverse, or you've got extra reactions. So that indicates that that constitutes a red cell discrepancy, and you have to figure out what's causing that. So the first and the easiest thing is to rule out some type of technical error. So was there an error in documentation? So we want to make sure we've got the correct patient sample identification on all of the tubes, verify the interpretations of the test results for accuracy and proper documentation. We want to check to make sure that something was not wrong with our reagents or our equipment. So check to make sure our daily QC on our ABO typing reagents, that those results were acceptable. Reinspect your reagents for any kind of contamination or hemolysis. Make sure that the reagents are in date. Confirm accurate centrifugation time and calibration dates. Was the standard operating procedure followed? So we want to verify our procedures follow the manufacturer's direction in the package insert. Did we use the appropriate reagents in the initial testing? Check our red cell suspensions to make sure they're concentrated correctly, and verify complete resuspension of the red cell button before we grade our reactions. So all of that is are quick and easy ways to initially try to resolve a discrepancy. If you've determined that all of that was appropriate, then you have to go on and determine where the error is at. So is it a sample area error? Have you got a discrepancy that affects the ABO red cell testing, which is our forward typing? So have we got extra, missing or weak, or mixed field reaction? So if it's an extra antigen that's present, it could be due to an acquired B or a BA person. It could be due to polyagglutination, which can happen in cases of cold antibodies. And in that case, you would see agglutination in all of your tubes in the initial spin or room temperature reaction phases. Is it rouleau? So you would see what appears to be agglutination in all of the tubes, but rouleau will disperse with the addition of saline, and rouleau looks, instead of looking like clumps, it'll look like the red cells are kind of stacked. Stem cell transplants, if the patient has had a stem cell transplant, they could start producing different cell populations. Okay, is it due to missing or weak antigens? It could be the result of an ABO subgroup, some type of pathologic etiology, so the patient has some type of underlying disease. Could be to transplantation. Okay, if you're seeing mixed field reactions, could be transfusion of large volumes, which is referred to as massive transfusion, especially of group O donor cells, which is what happens in cases of emergency release. So if you've got massive transfusion of group O donor cells to either group A, group B, or group AB patients, those group A donor cells are going to contain low levels of anti-A and anti-B, which can be passively acquired by the recipient and demonstrated in that patient's reverse type. It also, those massive transfusions can also dilute the patient's blood, so you might see weaker reactions because of that. Again, stem cell transplants and patients who are A3 phenotype. Okay, and then discrepancies that affect the plasma or reverse testing. If you've got presence of additional antibodies other than anti-A and anti-B, so anti-A1, which it's possible for A2 patients to produce if the patient's got cold alloantibodies. So these are red cell antibodies that are specific for other human red cell antigens that typically react at or below room temperature. So you would see reactivity in your initial spin reactions. Cold autoantibodies, so the patient has red cell antibodies specific for their own antigens, again, that are typically going to react at or below room temperature. And most of the time with autoantibodies, you're going to see reactivity in all of the cells. Rouleau, again, those are red cells that are stuck together. They look like a stack of coins. Or have you got missing or weak ABO antibody reactions? Could be due to the patient's age. Either they've not formed antibodies yet, in the case of very young patients, or they could have lower concentration of antibodies in older patients. Diagnosis, so reduced antibody levels are associated with different pathological states, such as A-gamma globulinemia, which is absence of the gamma globulins and antibodies in the blood due to leukemia, myeloma, or lymphoma. And has the patient been on any type of immunosuppressive therapy for transplantation? Okay, and here is a good chart that breaks down what you're seeing in either the forward, which is your red cell testing, or your reverse, which is your plasma or serum testing. So which are you seeing the reactivity in the forward or the reverse? And then what is it that you're seeing that is discrepant in that forward or reverse testing? And then, so if you're seeing extra antigens, this list here could be possible reasons why, and so on. Okay, so resolving some of the ABO discrepancies. So the first thing you have to do is rule out those technical errors. Most discrepancies are related to technical errors. So go back and check everything that you've done. Did you identify the patient properly? Did you use the correct reagents? Did QC work properly? So check all of that stuff first. If a technical error has not found, then you could repeat the ABO typing just to make sure it would exclude any kind of error that was that occurred during testing. And then if you're still seeing that discrepancy, get a new sample from the patient. Do it again. If the discrepancy remains after that point, after you've repeated the testing on the first sample, repeated the testing on a new sample, then you're going to start looking beyond that. So we're going to look at the patient's medical history to see if there's anything there that could indicate why there could be a discrepancy. And then we're going to start looking at where those reactions are occurring and what exactly is happening. One quick way to resolve an ABO discrepancy that you think might be due to Rouleau is replace the serum with an equal volume of saline and mix it, centrifuge, and then if you still don't see that what you thought was agglutination, it indicates that it was Rouleau. If you are still seeing agglutination, then it is true agglutination. Another way that this can happen without having to replace the serum with an equal volume and then centrifuging, just add a couple drops of saline to the sample and then look at it again under the microscope. And then again, here's that same chart again that just kind of breaks it down into what phase of testing is the discrepancy showing up at. So is it in the forward type or the red cell testing? Is it in the back type or the plasma testing? And all of the different things that could be attributed to if you're seeing extra antigens missing or weak antigens, mixed field reactions, what it could be due to. And there are some additional videos in Moodle on how to prepare a red cell suspension, the procedure for the ABO, RH, forward and reverse typing. So make sure you take a look at those before you come to class. And this is the end of Unit 2.